Good morning. So it's a great pleasure to have all of you for the Network Biology Day. So it was meant to be a very small meeting. A brief background how this meeting was envisaged. We started 10 minutes late. I must mention that we have accounted for such delays in our program, so I think we will finish on time today. So I'll just make some brief comments. I'm Arijit Samal. I'm a faculty in the computational biology group here. I'm one of the organizers. So the idea behind the meeting was as follows. So if you look at IMSC, we have roughly 5 faculty and about a community of 30 people now working in computational biology. This includes students, postdocs, project assistants. Similarly, if you look at IIT Madras across various departments, there are at least 10 faculty working in the area of computational biology. In particular, the biotech department has 7 faculty members in the area of computational biology. A subset of these faculty who are working in computational biology in the 2 institutions work particularly in applying network concepts in tackling biological problems. In addition to these academic institutions, if you look at, there are also companies which are R&D oriented, like Vantage Research, QBiome Research, which has representation here today, where people who have done active research and continue to do active research for societal problems using network concepts. So we try to represent all of these people in this meeting today, and I'm hoping that there will be much more interaction between the student and the postdoc community which has gathered. Although the audience currently is about 55 people, we expect to go up to 94 people because people will trickle through the day. So with that, what I would like to say is that I would like to request our director, Professor V. Ravindran, the director of IMSC, to deliver a short welcome address. Thank you, sir, for finding time. Please.
Thanks, Arjit. Good morning. It's indeed great pleasure to welcome all of you to our institute. On behalf of IMSC, I take this opportunity to welcome to IMSC, also to Chennai. I'm surprised that it's going to be only one day with so many people from all over India. I wish Arijit could have organized it for a longer period. I'm sure you will have an intense program today, and I wish you all have a very good time, good food, and also enjoy the ambience of IMSC. We have about 50 faculty members working in 4 different areas like physics, mathematics, computer science, and of course theoretical computation biology group. So do spend a few minutes of your time to go around the institute and get a feeling of what IMSC is actually, if you're going to be here for a day actually. Okay, so again I welcome you all. I won't take much time because it's already late by about 12 minutes, and let the business start. All the best. Yeah.
Thank you, sir. Thank you. Before we get to the first speaker, let me make some announcements. So this meeting would not have been possible without the support of the IMSC administration and the director. So to tell you the truth, we started planning this meeting on 4th July. So we have it on 16 days. So that should tell you the efficiency of IMSC administration. Actually, I can share an anecdote. When I had my offer from IMSC, I had met Professor Bhaskaran in ICTP, and I was considering. So he had mentioned to me, if you remember, in Adriatico guest house, that our administration is more like a Mercedes-Benz, while most of the Indian institutions' administration is more like ambassador car. So that was one of the compelling things he told me, which led me to accept this offer. So we, as an institution, are very efficient. We are a smallish institution, so we could do it in 15 days. So that's hats off to our director, who is support, and the registrar and the administration. So this won't have been possible without that. In addition, I would like to record a special thanks at the outset for our media team, the electrical team, and in particular the funding, because I know none of us discharged to anybody, because we are supporting this from the DAE EPICS project, funding to computational biology and other projects. So I express my gratitude to all of these people and the funding agencies at the outside that we are able to do it today. So as far as the program goes, we have 10 speakers, we have 15 poster presentations. You can see the coffee breaks and the tea breaks and the lunch and the high tea. They are all outside the auditorium in the portico. If you are attending the dinner, it will be on the other side on the first floor of the canteen. I will make further announcements down the line about various things through the day. So with that, what I would like to do is to introduce my first speaker, our first speaker, Professor Sitabro Sinha. He needs no introduction. He's a pioneer in the area of complex systems in the country. When I think of Sitabro today, I remember 2006 when I was a summer school student, summer school conducted by Sitabro. He has always been like a guru to me. I have never co-authored, but he has been an inspiration throughout my life. So I won't say beyond that. So he's an inspiration at various levels to me. So, and he also happens to be the Dean of Computational Biology Group here. So I would like to welcome Sitabro to give the talk and also request him to say a few words about the Computational Biology Group.
Thanks, Arijit, for that very kind intro. So let me extend a warm welcome to all of you on behalf of the Computational Biology Group at the Institute. So the group is actually the youngest of the 4 area groups in the Institute. As Professor Ravindran mentioned, we have physics, which is the oldest, followed by mathematics, theoretical computer science. And in 2012, several of us who joined the physics group originally but had strong interest in modeling biological systems decided that it would be good to have a separate graduate program where we can induct students having background not necessarily in physics. So, earlier most of our students would have physics or maybe applied maths backgrounds. But we were thinking that maybe if we have a separate graduate program, we could also get students having more of a bio background. So that was the genesis of this graduate program. So our university, which is the Homi Bhabha National Institute, the virtual university which is organized by the Department of Atomic Energy, very kindly agreed, and under the Biological Sciences Board of Studies, the Computational Biology graduate program was set up. So our first students were inducted, I think, around 2012 or so, and we have had several batches of students who have graduated since then, who are doing postdocs in various places in abroad and India. So within, as Arijit mentioned, there are 4 core faculty members and additional faculty member who are still part of physics, who constitute the group, and large number of students, postdocs, and project assistants. So within the group, a large range of subjects is spanned, ranging from network biology, as is obvious, to epidemiology, regulatory genomics, bioinformatics, physical biology, molecular dynamics, membranes, you name it, we have someone working on it. So I would definitely ask you, if you're new to the institute, to explore the various areas which are being worked on in this institute, and of course, as Professor Ravindran mentioned, do also look at the extra academic side, and please enjoy the atmosphere at IMSC. All right, so without further ado, let me get to the academic part of the session. So I'll be speaking about some recent work that my group has been doing. So this is primarily the work of my former PhD student, Anand Pathak, who's currently doing his postdoc in the US, and a senior scientist in our group, Shakti Menon, who is actually in the audience. So as the name makes it clear, it's something to do with brains, and so you might think of it as a computational neuroscience work, but actually the techniques that I'll be talking about go way beyond computational neuroscience and applies to network biology in general. It just so happens that we applied it primarily to connectomic data, because there are certain challenges in connectomic data which make it far more difficult to find hierarchical organization in them than in other kinds of biological data. So the fact that the method that I'll be talking about works so beautifully on connectomics data leads us to believe that it could also be used to find similar structural organization in networks spanning the range from cell to ecologies. All right. So, why connectomics? Because it's about the brain, and one thing that you ask the most clueless person on this planet about what they know about the brain, and the one thing I think they'll all agree is that it's complex. In fact, it's been mentioned that it's the most complicated kilogram of matter in the universe that we are aware of. So, where are we in terms of trying to understand it? Despite occasional puff pieces on the scientific magazines, I think we are kind of still far off. We still don't fully understand even this organism, the Caenorhabditis elegans, the first organism whose entire nervous system was mapped out. This is the pioneering work of, among others, Sidney Brenner, for which he won Nobel Prize, I think, sometime in the early 2000s. And it has only about 300 neurons, all right? And these are connected, with roughly 3,000 connections. So it's something, you can, in principle, map out on a piece of paper if you have enough patience, just with your hand. And we still don't quite understand how it works. So given that we don't quite understand how this works, it's probably too premature to say that we're anywhere close to understanding the brain that we are really interested in, which is our own brain, which, as you can see, has orders of magnitude more neurons than C. elegans, and each of these neurons in turn have about 1,000 connections, which eventually lead to about 10 raised to 14 connections. So it's complex. And what makes this complexity even more forbidding is that you have this complexity at various scales. So if you look at how people try to understand the brain, so someone says that they're a neuroscientist, you go and start talking to them, and you quickly realize that maybe your area of specialization has very little to say to that person's area of specialization. You may be working on completely different scales. So for example, you could have someone who's working at the molecular scale. Okay, so here we are talking about order of angstroms. Okay, there are lots of people who are working on synaptic communication, and here we are talking about micron scale activities. And people working on neurons, the motor transport in neurons, for example. So my colleague Sandeep, for example, works at this scale along with his collaborator Dr. Sanjay Kaushika. So that's at the scale of 100 microns. Okay? And then you look at networks of neurons. So spatially that's about a millimeter. And then large number of networks form maps of the external world to some kind of a neuronal encoding of them. So this encompasses regions of the brain which span about a centimeter, and then you have entire systems devoted to let's say processing visual information or processing auditory information. So that's a pretty large chunk of the brain, so that will be of the scale of 10 centimeter. Okay, and then finally you have the entire central nervous system, which essentially has nerves running all over our body, and so that's about the scale of a meter. Okay, and once of course you go to this level, you're talking about a functioning human being, and so you have the domain of cognitive science. So you'll have people like psychologists who are actually talking to this person who's having the brain, trying to figure out the internal states, the subjective experiences of the person, and so on. So what we are trying to do is trying to work somewhere at this scale. Okay? So we are neither at the molecular or synaptic scale or even the cellular scale, nor are we yet at this level where we are actually trying to work out what's the connection between the neuronal activity and the subjective experience of being conscious, but trying to see if we can map the activity of networks of, let's say, thousands or maybe even 1 million neurons to some behavior which you can actually observe. So this has sometimes been called the domain of systems neuroscience. Also, sometimes this is known as network neuroscience, because networks is the primary language which people use to talk about phenomena at this scale. So the particular question that we have been addressing over the last couple of decades is trying to find universal principles for describing how these nervous systems are structured. And what we are particularly interested, so what do we mean by universal? It means that are there principles which work across scales, which will work at the scale of a few neurons all the way to the scale of the entire human brain. Okay. So at one end, for example, you could be looking at network of, as I said, 300 neurons. So this is that worm, Caenorhabditis elegans, whose entire nervous system has been mapped out. So we know what are the neurons. So we know the names of every neuron, where they are located. We know which neuron talks to which neuron, so it's all genetically determined. So one of these worms, pretty much its nervous system looks pretty much same as another worm. So it's very convenient to study such a model organism, right? So whereas in humans, of course, our synaptic connections change because of experience. So twins who otherwise share the same genome will end up with very different brain organizations simply because they will have different experiences, right? So there's a huge variability. So fortunately for us, this model organism doesn't have that variability, so you can pretty much, study lots of different worms and try to work out how the structural plan of this nervous system allows it to survive in the wild, when our best efforts at creating robots and AI are still kind of going to fall flat the moment you put them in the real world. At the other end, we have the so-called macroconnectomes. So here we are not talking about network of neurons per se, because as I mentioned to you, humans have essentially close to a tenth of a trillion neurons. So there's no way we have the technology now to actually map out all the connections. So instead of that, what you could do is you look at brain regions. So brain regions, of course, come in different sizes. You could have brain regions comprising 1 million neurons. You could have brain regions comprising tens of thousands. You could have brain regions comprising, tens of millions of neurons. Traditionally, they have been identified by neuroanatomists from the 19th century. So, you have people like, for example, Paul Broca, who had worked out that there's a part of the brain which is responsible for language. It's known as Broca's area. And that part of the brain is, of course, always it's that specific area of the brain in any human brain which is responsible for language, and that's called Broca's area. So work of hundreds of neuroanatomists dating from the 19th century has allowed us to amass this information about the fact that the brain is organized into hundreds of different regions, each of which has a very specific location. So even though one human brain can differ in the detailed circuitry from another human brain, the area locations would be roughly the same. And so now we can ask, how are these areas connected to each other? So in this picture, essentially each of these nodes is not a neuron but a brain region. Okay, so they represent something like, it could be 1,000 or million, depending upon what's the size of that brain area. And these connections represent the so-called tracts. So you know that a neuron connects to another neuron with a chemical synapse or maybe an electrical gap junction. But now we're not talking about individual neurons, we're talking about brain areas. So what happens is that when you have 1,000 neurons over here wanting to talk to some of the thousands of neurons over here, you could of course send from this neuron a synapse to this neuron here, but then because you need to send so many connections, what do? You do what any self-respecting electrical engineer would do. Take all these individual wires, put it together, tape it together, put it in some kind of a plastic tubing and create a large cable. And that's exactly what the brain does. So they create these cables, and these are called these tracks, which will connect, let's say, this region to this region. And by using various staining techniques, you can actually identify these tracks. So again, for hundreds of years, anatomists have been using various kinds of tracing studies to map out all these connections. And today, we have been able to compile, and when I say we, I don't mean I personally or even my group, it's an international giant collective effort. They have put together all these hundreds of years worth of data to actually create a comprehensive map of these large brains. So for example, this is the brain of the macaque, which is this common monkey that we see all around us, a menace in the IIT campus and also occasionally in IMSC. So it's a primate brain which has been very well investigated. And for humans, fortunately we have a new technology by which we don't actually have to take the brain out of the dead organism and then do dissections and do tracing, which is how the macaque brain was studied. But for humans, you can't of course do that for ethical reasons. But fortunately for us, new technologies like diffusion tensor imaging has now been developed, by which you can look at a live individual, do imaging and actually construct these kinds of networks. So now you can have for each individual a particular structural network, and you can study what are the similarities and differences between siblings, the brains of siblings, for example. So the technology is there, the connectomic information is there, and now the question is, what do we do with this data? As Sidney Brenner once put it, we are in a situation where we are drowning in data, thirsting for knowledge. All right, so given this network, how do we characterize it? How do we extract information out of it? Okay. So for those of you who have already done courses in network analytics or network science, this would, of course, appear childishly simple to you. But for those of you who are encountering this for the first time, you can actually look at a network at various resolution scales. So you can look at micro scale, where you're focusing on local properties, and so there you are asking that, look, if I focus on a particular node, so this node could be a neuron, or this node could be a brain region, or of course if you are interested in something else like cell regulation or gene regulation or something, it could even be a gene or whatever. So we call that a node, and so you could be asking, how many connections does a node have? Okay. So that's one of the simplest questions, right? Or you could be asking, okay, I'm connected to so many other nodes. Are they also connected with each other? So is my neighborhood tightly clustered? Do they all know each other? Or do they all talk to each other? Or are they not actually talking to each other? They are each, kind of individual strangers and so on. I am the only one who's like common to all of them. Okay, so these questions essentially are the ones which people who study micro properties focus on. At the other scale, you could be looking at the network at the macro scale, and there you're asking questions about global properties. So for example, you can ask, how fast can I travel, let's say from this node to this node, if I'm only allowed to jump from one node to its neighboring node? When I say neighboring node, that means there's an actual explicit connection, let's say a synapse or a gap junction between these two nodes. So when you are sending information from one neuron to another neuron, the usual way is that the electrical activity of one neuron would travel through synaptic connection to some other neuron which has a direct synapse with it. And then from there it will go to another neuron which has a synaptic connection with the second neuron and so on. So if you have to travel through these synapses, you actually have to go from one neuron to a neuron which is connected to it, to another neuron which is connected to it. So the question is, how many neurons do you have to, intermediate neurons do you have to go through in order to go from, let's say, a sensory neuron here to a motor neuron here. By the way, this is not a neuronal network at all. It's actually a completely different, I think it's a protein-protein interaction network if I'm not mistaken. But this is just to illustrate to you the point that the questions I'm asking over here is like, what's the path length? What's the diameter of these network? Okay. What we are on the other hand focusing is at this scale. It's neither macro nor macro, and so we call it meso. Okay. So here we are looking at intermediate level structures. So what are intermediate level structures? Okay. So, and why do we actually look at mesoscale structures? So mesoscale structures allow us in some sense to do block modeling of complex systems. Okay. So we are taking a complex systems which is very hard to study in terms of its zillions of components. So what we could do is we could take the components which kind of hang together, and I would explain what I mean by hanging together, and then coarse grain them to form a block. Okay, and then we look at how these blocks connect to each other. So in a way, it's like when you're an engineer and you are trying to understand how a computer works. Instead of trying to look at each individual resistor, capacitor, or each individual electronic component, you would rather look at, for example, how does the control unit talk to the central memory store, how does it talk to its output devices, and so on. So you do this block modeling in order to not having to study this extremely complex machine at the scale of its most basic components. So that's the basic idea. So what are the kinds of structures? I mentioned examples of micro and macro scale properties. So what are the mesoscale properties that one could look at? So I mention here 3 of them. I will be focusing on this one, but this is also important because I'll mention a bit about it. So this pictures over here are schematic of what is known as a connection matrix that represents a neuron. So what's a connection matrix? So you can think of it as follows, that a connection matrix tells you how the nodes within a network are connected. So you have the various nodes arranged along the rows and along the columns, and so if you have a point over here, it tells you, is there a connection between this neuron over here and this neuron over here? So Aij refers to if neuron I and J are connected. So if Aij is 1, that means there's a connection between I and J. Now this schematic is basically saying that a modular network is represented by this kind of a block diagonal structure where you have very dense connections in these blocks and sparse connections in these off diagonal blocks. What does that mean? That means that you have groups of nodes, again this could be neurons or brain regions, which form communities which are very tightly knit. So you have lots and lots of connections within these groups and relatively sparse connections between groups. So this of course resembles our own social networks, right? So all of us have communities to which we belong, communities of friends, communities of relatives, and so on, and typically within communities you have very strong dense relations and you'll have a few connections across the community. Okay. So in that sense it's modular or a community level organization. Hierarchical on the other hand, as you can see, has exactly the opposite structure. So now the diagonal blocks have very sparse connectivity, and it's the off-diagonal blocks which have dense connectivity. So what does this mean? This means that now most of the connection is between one layer and another layer. Okay? So the network is now organized, instead of into modules, into layers. And most of the connectivity occurs between consecutive layers. So this resembles, for example, a corporate organization. So we do call corporate organizations hierarchies, for example, right? So what's happening is information is going from, let's say, the shop floor or the factory floor and is being refined. So you don't want to send, nitty-gritty details about, particular component missing to your CEO, right? So the bosses over here are deciding which information is relevant to be sent to their immediate superiors, and then those immediate superiors are deciding, okay, which information is worth sending it to my superior, and so on, until when it reaches the CEO has information only about the broad overall, happenings in the factory, so that she can decide what will be the plan for the next decade or so. Okay, and then she forms this, overall business plan, but not at the level where, a person at the shop floor knows, okay, exactly what I'm supposed to do tomorrow. So how to interpret that big business plan is the job of, these intermediary middle managers. And eventually when it comes to the person at the shop floor, it will essentially be decided in terms of, okay, this is what you're supposed to do tomorrow, the day after, this week, and so on. Okay. You can have yet another kind of mesoscopic organization. So this is called core periphery, where, part of the network is very tightly clustered. So everybody is talking to everybody, and this is also sometimes, likened with something known as a rich club. So everybody knows everybody over here, and then the rest of the people, the unfortunate ones, are kind of very loosely connected to this core. So I won't be talking about this at all, although we do have, work showing that nervous systems also have some aspects of this core periphery organization. Okay, so as I mentioned, modules are characterized by the existence of subnetworks which have very dense connection within them and relatively few connections between the modules. So does the brain have such an organization? And hierarchy, as I mentioned, is where you have a system where the nodes are arranged into various levels, and you have a very precise order in which these levels are placed one next to the other, so that connections are mostly occurring between successive layers. Okay. Now, of course, this would be very familiar to you from even your high school biology book. Okay, so for example, the usual high school biology book, when it talks about the nervous system, says that, the brain typically works like this. So the stimulus enters, for example, through one of the sensory organs like the eye, and the sensory neurons takes this information, passes it to the interneurons, and then eventually they're processed and sent to the motor neurons, where finally some action happens, like for example the cat chases the mice. And this broad idea has been borrowed by people working on artificial neural networks and today deep learning, where they actually have nodes arranged into layers, where they have an input layer which takes the, the stimuli, and they have multiple hidden layers which process this information, and finally of the output layer which gives this output. So the idea of this hierarchical structure is, in some sense deeply embedded in our intuitive notions about the brain. And the funniest part is, when it comes to actually figuring out what is the hierarchical structure of a real brain, we have no clue. Okay? So it's really interesting that although we all believe that the brain is hierarchical, when it comes to actually showing it, we are kind of clueless. So the problem why this is the case has been beautifully pointed out by the neuroscientist Sebastian Seung in his book Connectome, in which he says, look, the problem is this, that when you look at a connectome, it's looking like this. How do you disentangle to make it look like this, where the hierarchical arrangement is completely clear? Okay. Now, for this network, it's simple enough that you can do it by hand, but you can't do it for any reasonably large network. Okay. So how do you do it? Well, until this time, most neuroscientists who have been working on trying to uncover this hierarchy have been using additional information like functional information. What do these different neurons do? So typically neurons at different layers do different things. So if you know what the brain region or the neuron is doing, if you know that this is doing different stuff from this and this is directly using information from this, you can say, okay, maybe they belong to successive layers. Or you could use anatomical information. They may look different. Okay, so if they look different, they probably belong to different levels. And this is the way, for example, in which the hierarchy of visual areas of the macaque brain was reconstructed back in 1990s by Feldman and Van Essen. Okay, they used essentially anatomical and functional information. But the question is this: with this recent availability of large volumes of connectomics data, where we don't have this additional information about anatomy or function, we just have network matrices representing networks like this. How do you actually find this hierarchy? Can you find hierarchy from connection topology alone? Okay. To solve this, what Anand did was he took a leaf out of how do you actually find modules. So the problem of finding modules also is an equally difficult problem. How do you actually find subnetworks which are densely connected? So Newman, the network scientist, had suggested an interesting index. So he said that, look, suppose you look at the actual number of connections and you ask, okay, if the connections had happened by random chance, what's the probability that neurons I and J would be connected? And you look at the difference. Okay, and so you make some initial arbitrary determination that these are the modules. You find out this Q index. And then you ask that if I make a different choice of these modules, so instead of saying I is in module A, I say I is in module B and so on, would it give rise to a higher value of the modularity index? So then the question of finding modules simply becomes a question of maximizing Q by looking at different types of partitionings. And if it becomes a question of maximizing something, you can just use your favorite combinatorial optimization routine, like simulated annealing. So from a question of finding modules, it becomes a question of combinatorial optimization. So we had applied earlier this thing on finding the modular structure of the macaque brain, and indeed we found that it's actually made out of 5 densely connected communities which are very robust. So these are like the various realizations of the simulated annealing routine, which also beautifully fits with the functional purposes. So for example, one module is purely visual, one module is responsible for auditory processing, one module is responsible for olfaction and gustatory, so basically smell and taste. One module is responsible for somatosensory, motor coordination. Another module, interestingly, is not connected to stimuli but to emotional responses and formation of memory. So it was very interesting that here we found these modules purely on the basis of connectivity, and somehow out pops a purely functional segregation also. So clearly function and structure are very intimately related in the way the brain is organized. So how do we do it for hierarchy? So Anand had this brilliant idea. So he said that, look, given that hierarchy intuitively is defined as one where most of the connections occur between successive levels, why don't we define a hierarchy index where we say that, okay, let's do some arbitrary hierarchical determination where we don't really know what is the levels, but we make a random guess. We ask, okay, given the connections and given the random chance of connections between I and J, if they belong to successive layers, what is the value of this hierarchy index? And then we just try to maximize this H. Okay? So given this, taking an input which is completely scrambled like this, by doing maximization, and the maximization involves a number of refinement steps which I won't go into the details, you can actually get the final unscrambled hierarchical network. So it performs beautifully if you look at benchmark networks, so like synthetic networks which have both hierarchical and modular organization. So we got emboldened and decided to apply it to macaque. And we found that it finds this very robust hierarchical organization of 16 levels. So the colors represent, the various levels. So essentially, the 16th layer is over here, and the first layer is over here. So you can actually see how the connections go from layer 1 to layer 16, which is somewhere over here, because it's given in this kind of a plan, it might be difficult to work out. But by looking through this, we realize that there are actually two distinct streams along which information propagates along the brain. So there is a downstream pathway, which is, or rather, let me first talk about the upstream pathway. So the occipital lobe is where sensory information, like for example visual information, first comes in from the sensory organs. So there the information actually goes up to the temporal lobe and then go to the frontal lobe. So this is where you are doing higher level processing. But there's also a feedback path which tells, like for example, the eyes to move if you want to see something which you are interested in. So this is the downstream pathway where the frontal lobe gives information to the parietal lobe, which goes to the occipital lobe. So in the brain itself, this is how you have something like a circuitous feed forward and feedback loop. So what was really surprising was that it suggested that there is this organization which we call a modular hierarchy. So the brain is divided into modules. So we already mentioned that we had found all these modules, and we found these modules actually map very neatly into the various hierarchical layers. So each module has multiple hierarchical layers. That's one. And more interestingly, the modules themselves also have some kind of preferential arrangement with each other. So it's something where modules and hierarchy are intimately connected in a structural motif. So very qualitatively similar results were found for the human brain, for example. So here we had divided into 12 layers. Again, we find modular hierarchies there. And we also looked at multiple brains to show that very similar organizational plan is there across different individual human brains. This is for a 15-year-old, this is for a 27-year-old, this is for a 30-year-old, this is for a 42-year-old individual, and you can see broadly the similar hierarchical plan. And these are all for network of brain regions, so we decided to also look at network of neurons. And so for this, we went back to the Caenorhabditis elegans, and again we found that there's this very robust hierarchical organization of 12 layers. Interestingly, the sensory neurons are more numerous at the initial layers, and the motor neurons dominate the final layers, which is intuitively what you would expect. But remember, we are doing this purely from structural information alone. We don't have any functional information. And again, you have the modular hierarchy, where each of the 3 modules we found beautifully segregate into these different hierarchical structures. Okay, so the question is, why modular hierarchy? So this is where we are starting to speculate that, look, modularity and hierarchy actually have different functional roles. So modularity is needed when you want to do processing, when you want to process information which is specific for particular tasks. So when you are processing certain information relevant to vision, you don't want to immediately start processing information about something else. Let's say auditory stimulus. So initially you do all this information processing at separate locations. So that's when modularity comes in. But eventually you want to integrate all of this information. You want to know that this person wearing a red shirt is screaming like hell, right? So you want to integrate the auditory and visual information, and that's when hierarchy comes in, because hierarchy actually does sequential processing of information at different stages and then finally brings them together. And so functionally, modular hierarchy could be providing a basis for this systematic integration of information. And so to conclude, I want to just focus on 2 things. One is the algorithm that Anand and Shakti have developed very robustly uncovers this hierarchical organization which works across very different species. We showed it for human, macaque and C. elegans. We're pretty sure it will work for other species also. It's effective across scales, from the scale of brain region networks to neuronal networks. the nature of links, so whether it's directed or undirected, and the existence of other kinds of structural features like modules. Secondly, these results show that this debate between whether the brain information processing is sequential, as in a hierarchically layered system, or it's compartmentalized, as in a modular system, may be moot because the actual information processing could be done by structures like modular hierarchies, which combine aspect of both these organization principles. So I'm sorry for running out of time, so let me thank my collaborators, Shakti Menon, who is in the audience, and Anand, my former PhD student, who is now doing his postdoc at Dartmouth College, and of course, as always, very generous funding through the IMSS Center of Excellence in Complex System and Data Science, which is funded by the Department of Atomic Energy. So let me thank you for your attention, and thank Arijit for giving me this opportunity to present our work here. Thanks.
Thank you, Sitabro. Wonderful talk. So we are running slightly late, so but as a suggestion, we can have 2 questions now. I would request the speakers, if possible, to be present after their session, because in the breaks, so that if our audience wants to ask more questions, participants, they can approach them. So we can take 2 questions now. So whoever wants to ask questions.
So that was really wonderful. A very basic question. So in macaques versus human, as you were saying, there are regions for language, behavior, and other things. Are these conserved across species? So when you observe this modular nature or hierarchy in macaque, how well is it translatable to other systems?
Fantastic question. So actually, in our paper on the modular organization of macaques, Anand had pointed out that macaques have a very specific network motif precisely at the location where Broca's area is located in humans. So initially we were a bit worried about this because we were scared what the referees would say, because the moment you say that there is something like a pre-language area in macaques, they will say but primates don't have language, right? So, and we found that actually there's been a lot of literature over the last 10 years which is trying to show that there is something going on in that specific area which could be responsible for language-like phenomena, which is not language in the sense, the syntactic kind of structure that we are familiar with, but it is language-like. And so in some sense what we are seeing is the structural analog of that. So there is in fact a very close correspondence between the kind of subnetwork structure we are seeing in Broca's area in human brains and that corresponding area in the macaque brain. So there seems to be some kind of a species-species correspondence.
Questions?
I stop. So you said for the worm the number of hierarchy is more than for monkey. You said about 12 or I didn't.
Not sure. I mean it was 12, but I'm not sure. Yeah, so for this is 12. Let's say for primates it's
5.
It's 16.
I see.
Yeah.
So they are not very different from the worms.
Oh no, not at all, not at all. Like Nietzsche has famously said, right? We are the same as the worm, and to worm we shall return.
Really?
Yeah. He didn't know about salience.
So if there are no more further questions, let's thank Sitabro for this wonderful talk. Thank you, Sitabro.